trials can improve cancer patient survival, new drugs become available more quickly. So this is that we have, in the opening, we have to rush slowly.
Ladies and gentlemen, good morning. I am very pleased to have the opportunity to present the work I performed with Dan Turgeon here and Carla Bowman at Mayo Clinic, Rochester, Minnesota. With the advent of personalized medicine, we were wondering whether smaller, faster clinical trials could improve cancer patient survival. With increased knowledge of tumor biology, common cancers are more and more frequently recognized as consisting of small subsets of patients with particular tumor abnormalities. These abnormalities may be targeted by specific therapies. A good example is ALK inhibitors in non-small cell cancer. The issue is that in rare disease, it becomes really complicated to evaluate the many treatments that may be available for testing. A few words to define what we talk about. Randomized trials are clinical studies performed to demonstrate the efficacy of a new treatment, comparing the new treatment to the best available treatment, the so-called standard treatment or control treatment. Traditional trials are designed to avoid wrong decisions. In particular, we traditionally use stringent evidence criteria. We adopt the new treatment as the new standard, allowing for a very low false positive rate, what we call the alpha error. Consequently, many patients are required. The issue is that large sample size clinical trials take a very long time to reach a definitive result, especially if the disease is rare. So nowadays, by running a single trial over many years, we cannot evaluate the other drugs available meanwhile. So what happens if we reduce the sample size of clinical trials? If we relax the evidence criteria to adopt a new standard? To answer these two questions, we have simulated a series of clinical trials run over a 15-year research period, assuming, for example, 100 patients per year with various design parameters. We varied the number of trials run over the 15-year period, and consequently the trial sample size from 2 big trials to 10 smaller trials. We also varied the evidence criteria, the alpha error of the test. And then we measured the expected survival gain obtained at a 15-year research horizon. For example, we compare the expected gain at 15 years after 2 large randomized trials with stringent evidence criteria to the gain expected after running 7 successive trials over 15 years with relaxed evidence criteria. We observe that the expected gain is much higher with a strategy based on many smaller, faster trials with relaxed evidence criteria than with traditional designs. The downside of this approach is that by reducing the sample size of trials and relaxing the evidence criteria, we might select as a new temporary standard treatment, a treatment that does not work better than the existing best therapy. But we can demonstrate that there is a very low chance of a truly poor outcome. And also, the error could be quickly remedied if many drugs are successfully tested. Additionally, with new targeted agents, we think that there is likely fewer safety issues than with cytotoxic chemotherapy. For all these reasons, small backward steps can be judged acceptable if it is a price for a greater long-term benefit. In conclusion, in rare diseases, where there is no way to increase the trilateral rate, and if there are many drugs available for testing, the ability to test many of the promising agents available is hindered by the need to invest many resources into running a single trial for many years. Gain in cancer survival could be improved by running smaller trials with less stringent evidence criteria. But also by running smaller trials, a greater number of treatments can be tested and new drugs could become available more quickly. I thank you for your attention.